So I want you to take a second and just think about the billions of microscopic cells making up your body right now.
That's a pretty massive number to start with.
Yeah, right. But imagine if like every single one of those cells came equipped with a tiny built-in display screen.
A display screen. Okay.
Yeah, like instead of doctors having to guess what was happening deep inside your tissues, what if those cells were actively broadcasting their status?
Oh, wow. That's an interesting way to picture it.
Right, like a screen that literally reads, healthy, or maybe currently fighting an infection, or, most crucially, secretly developing cancer.
I mean, it sounds like a concept straight out of science fiction, really. The idea that our biology comes with this external status monitor just waiting to be read by anyone who knows the language.
Exactly. But according to the source material we're exploring in today's deep dive, this is actually moving out of the realm of science fiction.
Yeah, it really is.
We are looking at a May 18th, 2026 article from Science Daily, and it details some really significant research coming out of the Max Planck Institute for the Science of Light.
It's fascinating research.
So our mission for this deep dive is to, well, decode what scientists are calling a hidden sugar code on the surface of human cells. And we're going to explore how learning to read this code might just fundamentally change early disease detection.
What's fascinating here is that this research represents a major shift in how we view the relationship between the internal machinery of a cell and its outer environment.
Right.
It really changes our fundamental understanding of cellular communication.
Okay, let's unpack this a bit, because to really grasp the mechanics of this, I keep thinking about it like a smartphone, but not the way we usually think of a phone screen.
Okay, how so?
So imagine if your phone's protective case had a built-in e-ink display on the back, and the internal battery and processor were hardwired to constantly change the visual pattern on that case.
Oh, I see where you're going with this.
Yeah, so like if the processor started overheating or a virus hijacked the software, the physical pattern on the outside of the case would automatically shift to reflect that internal damage.
Right, so you get an external warning.
Exactly. You wouldn't need to crack the phone open to know it was broken. You could diagnose it just by looking at the back cover with, perfect mathematical accuracy.
That analogy tracks really closely with the biological reality we're discussing here.
Does it really?
Yeah, absolutely. I mean, the biological equivalent of that phone case has been known about for a long time, but it was largely viewed as, well,
Just uninteresting.
yeah, but to appreciate this breakthrough, we really have to look closely at what actually coats your cells.
Right. And the source refers to this layer as the glycocalyx.
Yes, the glycocalyx.
So before we get into how it communicates, we should probably establish what it actually is. For the listener, what exactly is the glycocalyx?
Well, every single human cell is covered by this very thin protective outer layer, and it is composed entirely of complex sugar molecules.
Okay, I should probably pause and ask a clarifying question here for you listening, and to be honest, to clarify my own mental image too.
Sure, go ahead.
When we use the word sugar in this context, are we talking about like the same chemical structure as the stuff I stirred into my morning coffee?
That's a very common misconception, actually.
Because when I hear sugar coating, I immediately picture like an M&M, a cell wrapped in a hard, crunchy candy shell.
Right, right.
But looking at the research from Professor Leonhard Muckel and his physical glycosciences group at the Max Planck Institute, it seems like a static shell is just the wrong way to visualize this.
Yeah, it's definitely not a static shell. I mean, they are sugars, yes, but we aren't talking about simple sucrose crystals like in your coffee.
Okay, so what are they?
In cellular biology, we're talking about glycans. These are incredibly complex, highly structured, branching carbohydrate molecules.
Branching molecules, so not a hard shell at all.
Not at all. This layer is highly dynamic. These sugar molecules are in a constant state of fluid motion.
Constant motion.
Yes, they continuously shift, they reorganize themselves, and they help the cell physically and chemically interact with its surroundings.
So they're actively responding to their environment, almost like a shifting mood ring.
That's a great way to put it, yes. For decades, the traditional view kind of relegated this sugar layer to just basic structural support or, physical barrier.
Like a biological bumper to keep cells from bumping into each other and getting damaged.
Exactly. But Professor Meckel's team is proving that the specific ever-changing arrangement of these sugars is actually a real-time reflection of the cell's internal biology.
Oh, wow. So the inside is directly talking to the outside.
Right. The cell's internal organelles are constantly synthesizing these sugars, modifying them, and then pushing them to the surface. It is actively displaying what is happening deep within its core.
Okay, here's where it gets really interesting to me. If these sugars are just, individual molecules and they're constantly shifting and reorganizing in this fluid state,
Right.
how do we actually read the display? I mean, we are talking about something unimaginably small. How do you take a clear picture of a screen that size?
And that, right there, is the core technical hurdle.
Mm-hmm.
I mean, knowing a code exists doesn't do you any good if you don't have an instrument sensitive enough to read the text.
Right, exactly.
And standard light microscopes, the kind you might remember from high school biology, they are fundamentally limited by the physical properties of light.
Because they just can't zoom in enough.
Basically, yes.
Yeah.
Because of something called the diffraction limit, they physically cannot focus on something as small as a single sugar molecule that just ends up looking like a blurry haze.
Okay. And the researchers solved this by developing a new technique they call Glycan Atlasing, which relies on super-resolution microscopy.
Yes. Super-resolution microscopy completely bypasses that traditional limit. It uses these special fluorescent molecules that act like tiny light bulbs.
Tiny light bulbs, how does that work?
So instead of trying to illuminate the whole cell all at once, researchers can attach these fluorescent tags to specific sugar molecules and then make them blink on and off, one by one.
Wait, really? Just one at a time?
Yeah, and the computer tracks exactly where each blink happened, pinpointing the specific coordinate of individual molecules.
Wow. So instead of a blurry photograph, you're building an exact topographical map, just dot by dot.
Exactly. And they didn't just map a general cell shape either. They mapped the glycocalyx across a massive variety of complex samples.
What kind of samples are we talking about?
Things like laboratory cell culture lines, primary human blood cells, and even actual human tissue samples. The precision of their mapping is actually why this work was published in the prestigious journal Nature Nanotechnology.
That makes sense. The visual they provided in the source material really helps ground this concept, I think.
Oh, the blood vessel cell image.
Yeah, they showed a map of the glycan structures on a human blood vessel cell. And, credit to researchers Dijo Munakondatil Joseph, Nazlikin Urekli, and Leonhard Mikkel for capturing this.
It's a stunning image.
It really is. When you look at it, it resembles a map of a galaxy. You see these sprawling, branching structures glowing in magenta, bright emerald greens.
Yes, and clusters of yellow and gold against that pitch black background.
Right. It is a highly detailed, unimaginably dense matrix of dots.
And the density you're describing is precisely what makes analyzing these structures so incredibly challenging. I mean, there are billions of data points interacting simultaneously.
So I look at that incredibly dense image, and I just have to ask about the methodology.
Okay.
Because mapping individual molecules across entire tissue samples sounds like, I don't know, trying to map the exact location of every single blade of grass in the Amazon rainforest.
This is a massive undertaking, definitely.
Right, and with that much biological movement, how do the researchers know they're seeing a meaningful pattern or real code and not just random biological noise?
If we connect this to the bigger picture, one of the fundamental principles of biology is that an organism rarely expends energy on purely random processes.
Okay, that makes sense. Energy is valuable.
Exactly. Maintaining and reorganizing millions of complex sugar molecules takes a significant amount of cellular energy. So if they are shifting, there is an underlying architectural rulebook.
So it's not just the wind blowing the leaves around, to use the rainforest analogy.
Right. When you can only see a low-resolution, blurry image of our hypothetical rainforest, a shifting canopy just looks like the wind. It looks like random noise.
Sure.
But when super-resolution microscopy allows you to zoom in and track the exact coordinates of every leaf over time, you realize the movement isn't the wind. It's a highly structured response.
Oh.
The researchers track the statistical distribution and the deliberate clustering of these molecules. They basically prove the shifting is highly choreographed.
Okay, so if we have the technology to map this molecular rainforest accurately, I guess the next logical step is comparison. What exactly happens when researchers take the map of a perfectly healthy cell and compare it to the map of a sick one?
Well, the researchers demonstrated that the molecular arrangement of the glycocalyx changes directly and predictably based on the condition of the cell.
Predictably.
Yes. They observed this particularly closely in immune cells, actually.
Oh, right. The article details how they watched immune cells change their sugar layouts when they were stimulated.
Yes, the activated versus inactive states.
Right, so an inactive resting immune cell has one specific pattern on its outer screen. But the moment it detects a threat and activates, the sugar pattern physically reorganizes into a completely different layout.
Exactly. And that reorganization likely helps the activated immune cell better adhere to pathogens or maybe communicate more effectively with other cells in the immune system.
That's amazing.
The outside of the cell is in a constant structural dialogue with its internal state. The glycocalyx acts exactly like that display screen you mentioned earlier.
But the finding that has, I think, the most profound implications for the future of medicine involves cancer.
Absolutely.
The researchers found that these nanoscale sugar patterns can reliably distinguish between separate stages of cancer development.
Which is huge for diagnostics.
Huge. They took actual human breast tissue and successfully distinguished the cancerous regions from the healthy regions just by reading the patterns on the glycocalyx.
Right. The sugar patterns on the malignant cells were fundamentally distinct from the healthy tissue surrounding them.
Now, from an evolutionary standpoint, that feels a little contradictory to me.
How so?
Well, if a cell is mutating and turning cancerous, its primary goal is survival, right? Survival and unchecked replication.
That's the nature of cancer, yes.
So why would a rogue cell broadcast its mutated state on its outer display screen? Wouldn't a cancer cell want to camouflage itself from the body's immune system rather than broadcasting, hey, I'm cancer?
That's a really great point.
Like-
And we shouldn't view this as a deliberate advertisement by the cancer cell.
Okay, so it's not doing it on purpose.
No, the cell isn't intentionally raising a red flag. It is actually an unavoidable physical consequence of how cancer functions.
An unavoidable consequence? How does that work?
Well, when a cell turns cancerous, its metabolism goes into absolute overdrive. It begins consuming massive amounts of glucose and other nutrients to fuel that rapid, unchecked division.
So it completely changes how it eats and processes energy.
Exactly. And the crucial link here is that those complex surface sugars, the glycans, are synthesized inside the cell using those exact same metabolic pathways.
Oh, I see.
Because the cancer has hijacked the internal machinery and thrown the metabolism into overdrive, the cell's internal factory literally starts producing the wrong building blocks. It alters the enzymes responsible for linking the sugars together.
Oh, wow. It's like a car factory that's had its internal assembly line randomly sped up by a computer virus.
That's a perfect analogy.
The factory doesn't want anyone to know it has a virus, but because the assembly line is moving way too fast and using all the wrong parts, it just starts rolling cars out the door with 3 wheels and the steering column attached to the roof.
Exactly.
The internal defect becomes totally obvious just by looking at the outside of the product.
That is the exact mechanism at play here. The cancer fundamentally disrupts the internal production line, and the corrupted glycocalyx is the outward proof of that disruption.
It just leaks out as an error code.
Right. It is physically impossible for the cell to maintain a healthy sugar coating when its internal metabolism is that damaged. And Professor Meckel's paper notes that because this altered biological information is a reliable physical consequence of the disease, it can actually be read using a standardized approach.
That's incredible.
The error code follows predictable rules, which is a massive leap for diagnostics.
So what does this all mean for the listener? I mean, finding a standardized way to read an error code that a cancer cell cannot hide is a huge leap. But if scientists can reliably read this code in a lab right now, what is the timeline for this technology actually reaching you?
That's always the big question.
Yeah, how long until this moves from a high-tech lab experiment to something that actually helps you during a routine medical checkup?
Well, moving from the laboratory bench to clinical application always requires clearing significant hurdles. But the Max Planck Institute has outlined a clear roadmap for scaling this technology.
The article does list a few immediate next steps. First, they say they're expanding the method to analyze additional target structures.
Right.
So they want to move beyond just one type of immune cell or one type of breast tissue to map the unique error codes for a broad spectrum of diseases.
Yes. And second, they are focusing heavily on automation.
Automation, right. And third, they want to conduct large-scale studies with significantly larger sample sizes.
Professor Muckel's ultimate vision here is to associate specific surface patterns with specific disease courses, and importantly, therapeutic responses.
Therapeutic responses.
Yes, allowing cell states to be detected early and objectively. And that word objectively is vital.
Why is that the vital part?
Because current diagnostic methods often rely on subjective interpretation. A radiologist looking at a slightly ambiguous shadow on an MRI, or a pathologist manually examining a tissue slide.
There's human error involved.
Exactly. Reading a mathematical distribution of molecules entirely removes human guesswork.
But the second step they listed, the automation, that seems like a massive bottleneck to me.
It's a challenge, yes.
Right now, this entire process requires cutting-edge super-resolution microscopy. That is highly specialized, incredibly expensive equipment that frankly requires a team of physicists to operate.
True.
That doesn't sound like the kind of machine sitting in my local neighborhood clinic.
Yeah.
How big of a hurdle is this to actually scale up for routine patient use?
This raises an important question about how medical technology evolves. The current equipment is indeed specialized and expensive. However, if we examine the historical trajectory of medical tech, what begins in a specialized research facility routinely evolves into a standard, automated clinical tool.
Okay, can you give an example?
Consider the trajectory of DNA sequencing. The Human Genome Project took over a decade, involved thousands of scientists, and cost billions of dollars just to sequence one single genome.
Right, I remember that.
Today, genomic sequencing is highly automated. It fits on a machine the size of a desktop printer, and it costs a fraction of the original price.
Wow. So the engineering catches up once the biological value is proven.
Precisely. Once the diagnostic value of glycan atlasing is fully validated in those large-scale studies, the biomedical engineering sector will heavily invest in making the hardware faster, cheaper, and more accessible.
That's very hopeful.
And beyond just finding the disease, the mention of therapeutic responses in Professor Meckel's vision highlights an even more immediate clinical application.
What does that look like in practice? Checking for a therapeutic response.
It means the future application of this technology isn't solely about initial diagnosis. It's about monitoring how a specific disease reacts to treatment in real time.
Real-time monitoring.
Right. If you are diagnosed with a tumor and begin a specific chemotherapy regimen, doctors currently have to wait weeks or even months and take multiple scans just to see if the tumor is actually shrinking.
That wait has to be agonizing.
It is. But with this technology, doctors could routinely sample the cells and read the sugar screen. If the chemotherapy is effectively repairing the internal cellular machinery, the surface sugar patterns will begin to shift back toward a healthy baseline almost immediately.
Oh, wow. And if the pattern doesn't change, the doctor knows right away that the drug isn't working for your specific tumor.
Correct. You can pivot to a different, more effective treatment without wasting critical months on a therapy that your cancer is resistant to.
That is unbelievable.
It brings a level of personalized, relentless objectivity to oncology that we just haven't had before.
We started this deep dive exploring a part of the human cell that, for decades, was largely overlooked. You know, a layer of complex carbohydrates viewed as little more than just a structural barrier.
A biological bumper, as you said.
Yeah. But through the work of Professor Muckle and his team and their development of Glycan Atlasing, we've learned that this glycocalyx is actually a dynamic communication matrix.
It really changes the paradigm of cellular biology. By applying super-resolution microscopy to these dynamic structures, the researchers have managed to map a terrain that was previously thought to be unmappable.
Right. We know now that immune cells physically alter their outer patterns the second they activate.
Right.
We understand the specific metabolic mechanism that causes cancer to inadvertently broadcast its corrupted internal state by, pushing an error code onto its surface sugars.
Yes, the leaky factory.
Exactly. And we are looking at a roadmap where this hidden sugar code can be read to catch devastating diseases at their earliest, most treatable stages.
And eventually, to monitor the efficacy of those treatments with molecular precision, entirely removing the trial and error from complex care.
It just forces you to think about your own body, your own biology, completely differently.
It definitely does.
Right now, as you listen to this, billions of your cells are actively shifting their sugar codes, synthesizing and reorganizing a complex nanoscale code to interact with the environment inside you.
It's happening right this second.
And it brings up a final thought I want to leave you with today. If we are just now learning to read this complex sugar code to diagnose diseases, how long will it be until we learn how to rewrite it?
Moving from observation to active manipulation.
Exactly. Could the future of medicine lie not in flooding the body with harsh chemicals to destroy sick cells, but simply in subtly reprogramming their outer sugar screens?
That is a profound thought.
If cancer relies on metabolic chaos to alter its coding, could we artificially rewrite the glycan code on a tumor cell so it displays a massive danger signal? Deliberately cueing your own immune system to recognize it and do the work for us.
using the body's own defense systems.
Right. The screen on the back of the phone case isn't just telling us the battery is dying. Eventually, we might be able to tap the screen to fix it.
Absolutely fascinating.
Thank you for joining us on this deep dive into the hidden architecture of your cells. Keep questioning the unseen processes happening around you and the microscopic patterns actively shifting right inside you.